Hello and welcome to this talk. This is part of the FSL course, this online talk part of the FSL course. What we're going to talk about today is how we can use the general linear model to model, well, in this particular talk, single session data. So what you're seeing here is an overview of the analysis process for a single subject. And what we have is we have the pre-processing, and I'm guessing that you probably already listened to some Mark Jenkinson's talks where he talks about motion correction, high-pass filtering, spatial smoothing, things like that. Then what we have here is we're going to talk about today how we build a model using the general linear model to describe and model our data and then ask interesting questions of our data, from our data, through this model. And once we've done that, we end up with a map of statistic map. So this is a statistic image where each voxel might be a t-value or an f-value, something like that. And then what we're doing is we're thresholding this to find out which part of the brain was significantly activated as part of our experiment. Now, I think personally that this is the most important part for you to learn and to get your head around. And the reason for this is that the pre-processing, yes, it is very important, obviously, but there's not so much you can do about it. You run MacFlirt to do the motion correction, you do some high-pass filtering, but there's not much leeway for you to change things here. Same here. Once you have your statistic image, there's only so much you can do in terms of doing this thresholding. Obviously it's important that you understand how it is done. It's important you understand why it is done, but there isn't really that much you can do to affect it. Whereas here, the modeling of the data to build up a good and sufficiently detailed model of your experiment in order to describe your data, that's where you can really make a difference, and it can make the difference between significant activations or not finding anything at all in your study. So I think this is an important talk. I hope that after the talk you will agree with me. Okay, so what I've done here is I have chosen to divide this talk into two parts, partly because it's a little bit weird for me to sit here and just talk to a computer screen, and I'm probably going to need a little rest after a while, but also for you to be able to sit down and think about the things you learned before you go on to the second part. So in this first part, what we're going to do is I'm going to show you an example experiment. We are going to use that example experiment to together build a model that describes the data from that experiment, and then we are going to ask interesting questions from our data through the model using something called T and F contrast. Now, after the break, for the second part of the talk, there will be a mention of null hypothesis testing. We're going to talk a little bit about the residuals. We're going to talk about thresholding, particularly multiple comparison correction. These things will be covered in more detail in another talk in the FSL course, the inference talk, but we need to bring them up here in order for you to have come across these concepts before you do the GLM practical. Right, so before we go into the talk proper, I'm just going to suggest an alternative way of thinking about your data. Now, presumably you have, as I said before, presumably you have been listening to Mark Jenkinson's talks. Presumably you've heard him describe the data as a smallish number of volumes, and this is obviously a very, very useful way of viewing your data when you're doing things like motion correction, because when you think of one volume and then you have another volume and you align the first volume, or the second volume to the first, and then you take the third volume, you align that to the first, and you do motion correction like that. Now, a different and alternative way of thinking about your data is to instead think of it as a large number of time series, each time series having maybe 100 or a couple of 100 time points. And for this particular talk, or the particular thing we're going to do in this talk, this is a very useful way of viewing your data. So try and think of the data as a large number of time series throughout this talk. Right, so what we're going to do is we are going to try and learn through example. And we're going to do that by me presenting to you an example experiment, and then, as the rest of the talk, we are going to then take that example experiment and try to build a model together to best describe that data, and then start asking some interesting questions of that data using T and F contrast. So the example experiment is an fMRI adaptation of a classical PET experiment. It's the word generation versus word shadowing experiments. And those of you who know your PET activation study history will recognize this as a paper from the Phil by Kathy Price. And what we have here is 3 types of events, and the way that this PET experiment has been modified in order to allow us to do this in the MR scanner is instead of overtly saying words, because that is always problematic in MR scanner, the volunteers or the subjects are instructed to just think these words in their head, not saying anything, just think the words in their head. So, as I said, 3 types of events. The first type of event we call word generation, and what that consists of having a screen, and on that screen a noun is presented. And prior to going into the scanner, the volunteer has been instructed that whenever he sees a noun on the screen, he is to generate a verb that goes with this. So, car, and the subject might, for example, generate the verb drive, and he thinks drive. He doesn't say it, he just thinks drive. Or he's presented with door, and the volunteer might think open. So this is the word generation task, and we call it a word generation task because you actually have to come up with a word yourself, and in this case it's a verb that you need to come up with. The second task, or the second type of event, is called word shadowing, and again, the volunteer, before going in the scanner, has been instructed that whenever he sees a verb on the screen, like in this case walk, all he or she is to do is to just think silently to him or herself that verb walk. Or read, the volunteer thinks read. The third type of event is something called a null event, and I'll mention later why null events are important, but what a null event is, it is simply an event that isn't there, an event that isn't happening. So the crosshair just stays on, and all the volunteer has to do is to try and stay awake. So there are some other things we need to know as well, and what we need to know, for example, is the ISI, or InterStimulus Interval. So what we have in this experiment is we have a 6-second InterStimulus interval. And what that means is that every 6 seconds, an event of one of these 3 types will be presented. So at time 0, we have an event, might be word generation, word shadowing, or null. At time 6 seconds, we will have an event, it might be word generation, word shadowing, or null. And they're presented in a random order, so neither we, before the experiment, nor the subject actually knows what is going to come next. Right, what I'm going to do now is we are actually going to play this experiment. We're going to play the start of this experiment, so you're going to see exactly the same thing as the healthy volunteer in the scanner experienced. So I'm going to start the experiment now, and you can see that we have a hair cross on the screen. Oops, there is a word shadowing event, read. Now there's a long break, so that must have been a null event. Word shadowing again, walk. Door opens, so there's a word generation event. Car drives, so there's a word generation event, etc., etc. And it goes on like this until we have had 24 events of each type. So now you know exactly what the data is like. So what we're going to do now is, together, we are going to build a model that we can use to model these data with. And we're going to use something called multiple regression or general linear model. And that's simply the tool by which we analyze data like this. So the task is now to build a model for that experiment. And a model consists of predicted responses. So what we need to do now is we need to come up with predictions for what we think might happen in the brain as a response to these different types of events. So we're going to start out with coming up with predicted responses to the word generation events. So what might that look like? Well, this is a start. I mean, it's not going to look like this, but this is a start. So what you see here is the first 100 seconds of the experiment, and we can see that after roughly 50 seconds or something like that is the first occurrence of a generation event. And then it's going to be another, I'm guessing 12 seconds or 18 seconds, and then we have the second event, and then another 12 seconds, and then we have the third event, and then after 6 seconds the fifth, etc., etc. So these are the times at which the different events occurred. But this is not how we predict that the brain is going to respond, or rather, this is not how we predict that the bold response in the brain is going to be to these events, because we know that the bold signal is not instantaneous. The bold signal is mediated through blood flow. So there's this viscous fluid that takes time to get into the brain, takes time to clear out of it. So we know that it's not instantaneous. So this might be roughly something like what we think that the neural response is to these events, but it's not going to be the bold response. So in order to figure out what the bold response is, we need something called the hemodynamic response function. And what the hemodynamic response function is, it is simply our postulated event, our postulated response in the brain to a single very brief event. And this is something that's been determined experimentally a long time ago by people. And what people did then was they simply would present a very brief visual stimulus, for example, and look in the visual cortex and see what happened in the visual cortex. Or they might present a very brief auditory stimulus like this, and then they look in the brain to see what the auditory stimulus looks like, what the auditory response looks like in the brain. And it turns out that it looks something like this. So it starts, here the event starts, and after a short period, it starts going up. After 5-6 seconds or so, it peaks and it starts going down. And over here, we are something like maybe 10 or 12 seconds, and then we have a slight little undershoot. And then after 15-20 seconds, the whole thing is gone, and essentially the response is finished in the brain. So, in order then to come up with a predicted response to a number of events with these timings, what we do is we take these timings, this stick function, or this train of sticks, and we convolve that with the HRF. And what a convolution is, it's simply a mathematical operation. It's a mathematical operation that operates on two functions, and the result is a third function. And when one of the function is as simple as this, i.e. as the train of sticks, then what the convolution is simply taking this HRF and placing one HRF with start at each and every one of these event times. So we got one HRF there, we got one HRF there, etc., etc. And then we're adding these up, and this is our predicted response. And obviously we need to do this for the entire time series. So this is what we predict that the response in the brain would be. This is what we predict that the response in the brain would be to the word generation events, right? So what that means that if we spot a time series in the brain like this, and we then compare this to our prediction for what the response to the word generation events would be, and we see, oh, that's a really good match. So then we can say to ourselves, okay, I think that this particular part of the brain, this particular voxel, this particular part of the brain is involved in word generation, or is interested in word generation. So what we do is we have our predicted responses, and when we check those predictions against the different parts of the brain, and we can conclude which parts of the brain are into word generation. Now, that was the word generation. We need to do now the same thing for the word shadowing event. So again, we are taking this train of stick functions. We convolve that with the HRF. So this is now the train of stick functions for the word shadowing event. And we end up with this prediction here. And again, same as before. If we now see a voxel in the brain that has a response over time that matches what we predict that the brain would respond to word shadowing with, we can say, well, okay, this is a part of brain that is into word shadowing, or that's interested in word shadowing. So what we're doing, or what we're attempting to do, is we want to take these predicted responses that we've built now, the word generation response, the word shadowing response, and we're going to use these to try and fit the observed data as well as possible. And by fitting, what I mean by that is what we have here is we have the observed data, so this is this observed. The word generation regressor, we made that ourselves, so this is known. Word shadowing regressor, this is known. And then we have two unknown parameters, and you can think of these as the parameters of this parameter here, that is essentially telling us how interested this particular voxel in the brain is in word generation, and this parameter here that tells us how interested this particular voxel is in word shadowing. And what we're trying to do now is we're trying to find these unknown parameters so that this linear combination, i.e. beta 1 times this regressor plus beta 2 times this regressor, fits the observed data as well as possible. Right. Now, before we go on to explain precisely how that is done, what we're going to do now is I'm just going to have a slight detour to explain how we make these regressors. So we start out by these event timings, i.e. this train of stick functions, at high resolution. So we have a very long vector with high temporal resolution. So maybe we have 1 point here per 0.1 seconds or something like that. And we need to do that because we need to have very high accuracy in the timing of these events, or in how we place these events. Because if you remember, we had an ISI of 6, so they come every 6 seconds, or that's how close they come. But then we have a TR, we have a repetition time of the other experiment, and that might be 3 or 4 seconds. But in order to get this timing right, we must have a higher temporal resolution first. So we have this high temporal resolution vector that we convolve with the HRF, and we get our predictions at a high resolution. So this is our predictions of what's going on in the brain with a high temporal resolution. But then what we need to put into the model is a regressor that has the same number of time points, or the same number of points as our measured time series. So what we do then is we take this predictions at high resolution, and then we simply subsample that at the repetition time of the experiment, i.e. for each repetition time of the experiment, we go in and we sample this, and that becomes our regressor. Now, so the estimation part, finding the best parameter values. And what that means is we want to find the parameters beta 1 and beta 2, such that the linear combination beta 1 times this plus beta 2 times that matches our data as well as possible. So let's just play with this a little bit first to see what we can do. So let's say that we postulate that beta 1 is 0.5 and beta 2 is 0.5. So 0.5 times that plus 0.5 times that becomes this. And we can see, okay, that doesn't fit our data particularly well at all. Okay, so 0.5 and 0.5, they are not good candidates for beta 1 and beta 2. Can we come up with better candidates? Well, let's try setting beta 1 to 0 and beta 2 to 1. And it turns out that that is even worse. So clearly those are not good candidates either. But if we say that beta 1 is 1.04 and beta 2 is 0 minus 0.1, essentially 0, then we come up, we can get a very good fit to the data. So obviously this is not how it's done. This is actually a linear operation, the task of finding beta 1 and beta 2 such that this linear combination matches the data as well as possible. It's a linear operation. It's done very quickly for you by feet, but this is the principle. The principle is we're trying to find the beta 1 and the beta 2 such that this linear combination matches the data as well as possible. and we're matching the data as well as possible. What we mean by that is we want to minimize the sum of squared differences between our model prediction in blue and the actual data here in red. So this voxel here has this time series, and we can see that the best parameter estimate is that beta 1 is 1.04, beta 2 is essentially 0, and what that tells us is that this voxel is obviously interested in word generation, but not so much in word shadowing, actually probably not at all in word shadowing. We have a different part of our brain, completely different time series, but again you can see that even though the time series is completely different, we are able to fit our model to the data with, just as well as the first one, but now we have different parameter values. Now we have beta 1 1.10 and beta 2 1.02. So we need to have both of those in order to be able to match the data, and what that tells us is that, okay, this particular voxel, he's interested in word generation, and he's also interested in word shadowing. We have a third voxel over here, and what we can see here is that, and these are actually the best parameters we could find for this one, and that's essentially 0 for beta 1, essentially 0 for beta 2, and we can see that, well, we're not actually capturing any of the variance in this voxel, and what we can conclude is that, well, obviously this voxel isn't particularly interested in words at all. Right, so, and this, I have to say, when I first came across the general linear model for this kind of purpose a long time ago, and I didn't know much about stats, I didn't know much about anything really, but it blew my mind. It really, really blew my mind. I thought it was just fantastic. The fact that we have this very simple model, right? This very simple model, and yet we are able to model all these different time series simply by virtue of having different values for beta 1 and beta 2 in the different voxels, right? So what we have here is we have one voxel here, and we see we can fit that data very well using a beta value beta 1 of 1 and beta 2 of roughly 1 as well. This voxel here, again, we can model very well using a beta 1 value of 1 and beta 2 value of essentially 0. And this voxel over here, again, we can model it reasonably well just by simply having 0, 0, right? And the way this is estimated and the way this is stored by feet is that it's calculating these values, and so for each voxel here, it calculates a value for beta 1 and it stores that in a beta 1 map at that voxel location. And then we have this voxel location here, we have another value for beta 1 and it stores that in the beta 1 parameter map for that location. And we have another voxel here, we have another value for beta 1 and it's stored there in the beta 1 map, and the same for the beta 2 map. And if you're interested in where they are, these are in the feed directory /stats/PE1, so those are the beta values for the first regressor of your model, and /stats PE2, and those are the beta values for your second regressor of your model. And if you have more regressors, you can have PE3, PE4, PE5, etc. So we have now taken our time series and we have reduced the information in that time series down to essentially consisting of a model and two maps of parameter values that tells us how much is needed of each of those regressors in each and every voxel. And you can see already here in the beta 1 map. And remember, the beta 1 corresponds to how much we need of the word generation regressor. And we can see already here that we have a hotspot here on the left side. Well, it actually looks like it's right side, but it's the left side of the brain, which is clearly interested in word generation. Now, in addition to this, what we can do is we can also estimate the residual error. And the residual error is the difference between the data and the best fit. So what we have here in red is the data. The best fit is this blue line here, and the residual error, that is what we get if we subtract the blue line from the red line, and it's this one here. And the magnitude of these errors here is a measure of the residual error. So this is essentially the standard deviation of the error. So this is something we can also estimate, and it sort of comes out in the estimation process, just like beta 1 and beta 2. And again, we can just have one image where we write in. So what we're doing now is for each and every time series, each and every voxel, we are fitting the data, we're fitting the model to the data, and we get a beta 1 value, we get a beta 2 value, but we also get an estimate of the residual error, and that residual error is written into something called, or a file called /stats/sigma squared. So this is the standard deviation of the error, right? So this is now, so what we've done now is we built the model. Yeah, okay. So let's just go to the summary. So what we've done now is we built the model. The model consists of a set of regressors, i.e. tentative time series that we expect to see as a response to our stimulus. And the model, when we're building the model, as you saw, this is what we did, it typically consists of our stimulus function convolved by the HRF, the hemodynamic response function. And what the estimation does is finds the parameter values such that the linear combination of regressors best fits the data. Every voxel has its unique parameter values, and that's how a single model can fit so many different time series. And we can also get an estimate of an error through the residuals. Right. Okay, just formalizing this a little bit. So this is placed into the general linear model framework. And this is the equation that you will typically see not only in the modeling papers, but also in application papers and what have you. And that says that y, which is a vector y, so it's going to be boldface non-capital letter. So that's the data, and it says, okay, that is the design matrix, and design matrix is simply the concatenation of these different predicted responses multiplied by the beta values, or the regression parameters, or the effect sizes, how much we need of each and every one of those, plus the residual error, Gaussian noise. Okay, so this is just formalizing exactly what you have already learned. Right, so this particular slide is for people who come from SPM to FSL. So I started out in SPM myself, nothing wrong with that. But a confusing thing when you go from SPM to FSL is that there is no constant in the model. So in SPM, you will have a constant in the model, and that's just modeling the average of the time series over time. And we don't do that in FSL, and there are simply two equivalent options. So the mean value for an Fmri time series is completely uninteresting. So what we are interested in is the variance in that time series, and in particular the variance in that time series that is caused or introduced by our particular experiment. So the mean value is of no interest. So there are two things you can do. You can simply calculate the mean of each time series and just subtract that away from the time series, and then you don't need to model it, and that's what FSL does. Or you can put a term into the model to account for the mean, and that's what SPM does. Now, I'm saying that the mean is uninteresting, and mean is uninteresting at the first level when we're modeling time series. At the second level, I mean, you haven't come across it yet, but there will be a talk about second level modeling as well. There, the mean is interesting. So, for example, this is a model where the first regressor, so this is now a model where you have two different groups. So this could be healthy volunteers, this could be patients of some sort. And the first regressor here models the difference in activation between the two groups, which is obviously interesting. The second one here now, which is the mean, now models the average activation across the two groups, which is also interesting. We are interested to see, is there an average activation somewhere in the brain that is common to the two groups? So at the second level, the mean is interesting, or is sometimes interesting, so there we model it. At the first level, we don't model it. Right, now we're getting to T and F contrasts. And what T and F contrasts are, it's simply a way of asking questions of our data through the parameters of our model. And we're doing this using something called Cope contrast or parameter estimate. And what that is simply is a linear combination of the parameter estimates. And you will typically see them like this. So there could be 1, 0. And what that means is simply that this Cope, or this contrast or parameter estimate, is 1 times beta 1 plus 0 times beta 2, i.e. it is our estimate of beta 1. And if we put something like this in, we are simply interested in asking the question, is beta 1 0 or different from 0 in this particular voxel? And for the experiment that we have looked at, if it is different from 0, that would indicate that this voxel is interested in word generation. Or it could look like this, 1 minus 1, simply again, 1 times our estimate of beta 1 plus minus 1, i.e. minus 1 times our estimate of beta 2. And what we're asking there is there a difference between beta 1 and beta 2 in this particular voxel? Because if they are different, then this subtraction here is going to be different from 0. And what we're answering there is this a voxel that is more interested in word generation than it is in word shadowing? And this we can use to create a t-statistic. And what a t-statistic is, it is simply this Cope, i.e. this linear combination of the parameter estimate, divided by the standard deviation of this Cope, the standard error of this Cope. So now this part here, the standard deviation of the Cope, so you've seen what the Cope is. That is simply the linear combination of the parameter estimate. The standard deviation of the Cope depends on various things. It depends on the model. And if you know the t-statistic, you know that if you have a lot of measurements, then the t-statistic goes up, i.e. you are more confident in your statistical values. So a model which is a long time series would increase the t-value, whereas a model that is a short time series, a very short experiment, would decrease our t-value. It depends on the contrast in a slightly complicated way that we're not going to go into, and it depends on the residual error. I.e. if we have a large residual error, it means that our model doesn't fit the data particularly well in that voxel, and then we have a small t-value. If we have a small residual error, it means that our model fits the data very well in that voxel, and we get a large t-value, and we say that we have a high confidence in that value. Or we can look at it like this. So from the perspective of a given contrast, this part here is constant, i.e. it is the same in each and every voxel. Right? Whereas this one is different in each and every voxel. So you can think of the t-contrast as simply the contrast of parameter estimate divided by some constant that is the same for each and every voxel, multiplied by this map of the residual error. So what this one happens to be the Cope 1, 0, i.e. it just looks at, so this is just the map of the beta 1 parameter estimates, divided by a constant multiplied by the residual error map, and this is the t-map. And if you squint a little bit, you can see that it looks like we actually have something going on in an interesting part on the left side of the brain. Right, so let's go into the t-contrast. So if we look at these two t-contrasts, we take this as an example, 1, 0, so i.e. we are looking only at the first parameter estimate, and what that means is we are looking at generation versus rest, i.e. we are looking at the voxels that are interested in word generation. Or conversely, if we're looking at 0, 1, we're looking only at the parameter estimate for the second regressor, the regressor for word shadowing, i.e. we are looking at word shadowing versus rest. So let's have a little look here. So what we have here is a contrast weight vector 1, 0, and we have a particular time series here where we have a large value for beta 1 and a small value for beta 2. So this contrast weight vector now asks the questions, where do we need this regressor to model the data? i.e. what part of the brains are used when we see nouns and generate related verbs? So to generate our generate our t-contrast here, first of all we generate our contrast of parameter estimate, and the contrast of parameter estimate is 1, 0. So it is 1 times beta 1, i.e. the parameter estimate is the first regressor, plus 0 times beta 2, i.e. it is simply beta 1. So the contrast of parameter estimates in this case is exactly the same as the beta 1 map. And then to turn this into a t-map, we then divide that by the standard deviation of the cope, and if you remember correctly, that is simply a constant multiplied by our residual error map or standard deviation map. And this is the t-contrast in this case. So you can see that the t-contrast, it appears to be slightly noisier than the cope map, and this is because we have an estimate here that has some uncertainty in it, but we also have an estimate here that has some uncertainty in it, and the t-map is going to be a combination, or is going to have an uncertainty that is given by both the uncertainty in the cope map and in the error map. Right, so we can also do another t-contrast. We can look at the mean activation, so that would be the contrast 1, 1. So what we're doing there, we're simply looking at, just the average of the two. So let's go and have a look at that. So we have now we're looking at a different part of the brain. So we have a contrast wave vector 1, 1. So that means that the cope, the contrast and parameter estimates, is 1 times beta 1 plus 1 times beta 2, i.e. it is the addition of beta 1 and beta 2 that gives us the cope. And as before, the t-map is simply the cope, i.e. the addition of these two divided by the standard deviation of the cope, which is a scaled version of this map here, and it is this map. And again, we can see that something is going on here. So there's a part of the brain here that is interested in both word generation and word shadowing, since we're having reasonably large values for both. Now we come to some interesting ones here. So now we have a contrast here, a contrast weight vector -1, 1. So yeah, so what that is, it is simply the second parameter estimate minus the first parameter estimate. I were asking the question, what part of the brains are more activated or more interested in shadowing than generation? And in a similar way, we have the contrast weight vector 1, -1, and we're looking for the areas that are more activated by generation than shadowing. So let's go to our example again. So we have here a voxel, and in that voxel the value for beta 1 is 1.04 and the value for beta 2 is -0.10. We have the contrast weight vector that are looking for areas where the parameter estimate for beta 1 is larger than the parameter estimate for beta 2, i.e. areas that are more interested in word generation than word shadowing. So the cope is 1 times 1.04 minus, well, this, and we end up with 0.14 here, and the cope is simply beta 1 minus beta 2, and the t-map is the cope divided by the standard deviation of the cope as before. Now, if you're looking at this looks a lot like the 1, 0 map. Essentially, it's indistinguishable from that. So what does this new contrast 1, -1 give us over and above the contrast that we looked at earlier, the 1, 0 contrast? Well, let's have a look at this particular part of the brain that we've also looked at before, which seems to be interested in both word generation and word shadowing, because it has similar values for both beta 1 and beta 2. So if we look at the first contrast, we look at the 1, 0, i.e. we're asking the question, which part of the brain are interested in word generation? We can see that we get an activation in this area here, because clearly this part of the brain is interested in word generation. But if we are asking the question, which parts of the brain are more interested in word generation than in word shadowing, well, that's a different matter, because then we're taking the word generation map, subtract away the word shadowing map, and we see that we have no longer an activation there. So it's a difference between asking the question what part of the brain is interested in word generation and asking the question which part of the brain is more interested in word generation than in word shadowing, i.e. this is a more specific question. We're asking specifically, are you interested in word generation rather than what you're interested in here, which is a more general question. Are you interested in words in general? Right. So F contrast. So what we have, so this is a different way of asking questions from your data. And I'm going to try and explain this as well as possible. So again, we have two conditions, word generation and word shadowing. And what we want to know here now, is there an activation to any condition? Right. So now we are no longer interested in sort of making our question more specific. We are interested in asking the very general question, is there an activation to any condition, or either of these conditions? So first we ask, is there an activation to generation? And the question, is there an activation to generation? That's the 1, 0 T contrast. And then we ask, is there an activation to shadowing? And the shadowing T contrast, that's 0, 1. So we're having the word generation, the word shadowing T contrast there. And then we add the OR, i.e. we are ticking these boxes here to say, well, now we want to turn these into an F contrast. So we're adding, so the first one asks, which part is interested in word generation. Second one asks what part is interested in word shadowing. But then by ticking these, we add the OR. So which part is interested in word generation and or shadowing, because it's actually an and or in this case. Right. And that, asking that question, is equivalent to asking the question, does any regressor explain the variance in the data? So let's have a look at how this is actually done. Right. So we have our model, the model we've seen all along, and we have our data. And what we're doing is we are fitting our model to the data, and we're getting the residuals. This is exactly the same thing as you've seen before. But now what we do is we introduce a reduced model. And what the reduced model is simply the full model, but where we have removed any column that has a 1 in the F contrast. So you can see this column here has a 1 in the F contrast, so we have to remove this column. This column here has a 1 in the F contrast, so we remove this column again. So the reduced model is simply no model at all. So now we're fitting this reduced model, which is there's no model at all, and we're getting a different residual error. And what the F contrast is the difference between the residuals from the reduced model and the residuals from the full model, and then scaled by the residuals of the full model. And this is just to make it scale free. And if you think about this, it's quite intuitively clear that if these two regressors are able to explain a lot of variance in the data, then there is going to be a big difference in the explained variance depending on if you have them or not. Conversely, if this model had not been able to explain the data particularly well at all, then having them or not having them would not have made any difference, and there wouldn't have been a large difference. So you can see that the F value will be large when the model, or the part of the model that is spanned by the F contrast, is able to explain a substantial part of the variance in the data. And if you think about the question we asked, what part of the brain is interested in generation and or shadowing? That's exactly what we're asking. We're asking, so what parts of the brain can we explain the variance using this model consisting of generation and shadowing? So again, just look. So we have the full model, we have the reduced model, we have the sum of squared from the reduced model minus the sum of squared for the full model. And again, you can see here that the sum of squared looks to be larger here compared to here. And when we subtract this from this, yes, we can see this area here. And again, we see the same area as before activated as a consequence of Hodge. So F contrast, two conditions A and B, is any condition significant? Is any or both or one of them significant? And what we're doing is we are creating this F contrast by combining T contrast by clicking these tick boxes in the F contrast box. Right. So, summary. The general linear model is used to summarize data in a few parameters that are pertinent to experiment. The general linear model predicts how the bowl activity might change as a result of the experiment. And we can look for significant effects or differences by using T or F contrast on the GLM parameters. And this summarizes the first part of the talk, and tune in soon again to just listen to the second part of the talk where we're going to talk about the null hypothesis and the multiple comparison problem.